Hello everyone, I'm Mustafa Keskinler and I'm here with Teoman Çoruk, Mustafa Selçuk Erdinç, and Efe Uslu. We will be talking about CRISPR-enhanced microbial bioremediation for oil spill cleanup. We would like to start by addressing a very real and persistent problem, marine oil spills. These spills release massive quantities of crude oil into marine ecosystems. They impact catastrophic damage to biodiversity, long-term water contamination, and even disruption to food chains and coastal economies. Traditional cleanup methods, such as skimming, dispersants, and in situ burning, are not only slow and expensive, but in many cases, they create secondary pollution. Chemical dispersants, for example, break oil into smaller droplets, but often introduce their own toxicity to marine life. So, what's the alternative? Nature has already given us part of the answer: microbes. Among them, a marine bacterium called Alconivorax borkumensis stands out. It's known to naturally degrade hydrocarbons, the main components of crude oil. However, there's a catch. It only thrives under specific conditions: moderate salinity, nutrient rich water, and warm temperatures. Its hydrocarbon degrading capacity is limited to medium length chains, and its efficiency drops in extreme conditions. This raises a key question: can we make this natural cleanup agent more resilient and efficient so that it performs under real world marine stress? That's where our project comes in. We aim to enhance the capabilities of this bacterium using targeted genetic engineering. It's all for me. Selçuk?
Hello, I'm Selçuk Erdinç. I'm going to talk about our proposal and CRISPR editing plan. Our goal is to improve the resilience and oil degrading ability of A. borkumensis by editing its genome using CRISPR-Cas9. CRISPR-Cas9 is a revolutionary gene editing tool that allows us to make precise edits in an organism's DNA. We propose using it to upregulate two specific genes. The first is ALKB, which encodes for alkane hydroxylase. This enzyme catalyzes the initial step in breaking down alkanes found in crude oil. Enhancing ALKB may allow the bacterium to target a wider range of hydrocarbons, including heavier fractions. The second is DNAK, a heat shock protein gene that helps the bacterium survive in a high salinity, low nutrients, or temperature variable environments. By enhancing this gene's activity, we expect better survival and consistent performance. Importantly, we aren't adding foreign DNA, just modifying the bacterium's existing genome. This makes the changes more biosafe and acceptable under regulatory standards. That's all for me, Teoman.
Thank you. And I'm going to talk about the timeline, testing strategy, and the technical tools. Our implementation is broken into 3 years. In the first year, there will be a literature review, CRISPR design, and lab setup. We will select optimal CRISPR vectors like PCAS9 plasmids and design guide RNAs using bioinformatics tools like benchling. In the second year, gene editing begins. We will introduce CRISPR constructs into A. borkumensis using electroporation or conjugation. Successful edits will be verified using PCR and DNA sequencing. Modified strains will then be cultured in artificial seawater with controlled oil concentrations. Degradation will be measured using GCMS and growth will be monitored through OD600. We will also test stress resilience by varying salinity and nutrition conditions. In the third and last year, there will be a large-scale lab trials comparing wild-type and edited strains. Parameters like degradation rates, biomass, survivability, and biofilm formation will be statistically analyzed. Our estimated budget is under $1 million. It includes lab materials, gene synthesis budget, and equipment. That's all from me. Efe?
Thank you. This project has the potential to reshape how we respond to oil spills. Instead of relying on chemical dispersants or slow mechanical methods, we offer a bio-based alternative. Enhanced bacteria designed to work more effectively in ocean conditions. Of course, genetic modifications comes with concerns. That's why we propose thorough biosafety checks. We'll conduct whole genome sequencing to detect off-target effects. Additionally, we'll explore containment strategies like oxotropy or kill switch systems to prevent uncontrolled spread outside the lab. Why does this project matter? Oil spills remain a critical environmental threat that demands faster, safer, and more sustainable solutions. While nature provides us with microbial agents like Alcanivorex borchimensis, their effectiveness is limited by environmental stress and narrow substance range. Our project proposes a strategic enhancement of this bacterium through CRISPR-Cas9, aiming to create a more resilient, efficient strain capable of performing in real-world marine conditions. This project is more than a scientific experiment. It is a step toward practical, scalable bioremediation. It merges cutting-edge synthetic biology with environmental engineering to provide a non-toxic, genetically safe tool for marine restoration. By targeting a specific problem with a precise and responsible solution, we hope to contribute to a cleaner ocean and a more forward-thinking approach to ecological disaster response. Thank you for listening.